What's going on, family and friends and family? Welcome back here to Building Stuff with Nova. I'm excited today because we got an expanded building committee, as we affectionately call people who join us every day on Twitch, because we're not only on Twitch today, we are also on YouTube and we're on Facebook. Shout out to everybody here. Let me know where you're joining me in the chat, because today we have an exciting episode of Building Stuff with Nova. In case you didn't know, Building Stuff with Nova is like what we call your virtual makerspace, your ideation laboratory, your mastermind. And I am your, personal guide, your companion, your compadre. Sometimes I'm your mentor. Other times I'm your mentee. It all depends on what you're doing, or what we're doing rather. And you can call me Dr. Nee. Dr. Nehemiah Mabry is my name. And I'm happy to have you with me. So like I said, whether you're joining us on Twitch, shout out to our regular audience on Twitch. If you're on YouTube, or if you're on Facebook, I need to know where you're joining us from in the chat. I'm going to keep an eye on that right now to see who we have with us today on Building Stuff with Nova. I see Nova. Nova is always here with us, of course, because that's who we're building with. Nova Building Stuff, giving a nice warm welcome to everyone. We're, of course, kick this off. I see DuckTales, the cool cat himself, Mr. DuckTales, joining us. Good to see everybody here. I'm telling y'all, this is a great day. This is a great day because we have a guest. Today is a guest day, and I'm going to share with you all our agenda here in just a second. But yesterday, let me talk about yesterday. To our audiences on YouTube, audiences on Facebook, what we've been doing is going live every day on Twitch. Twitch knows this at 1 p.m. Eastern, and we've been engaging engineering. We've done it in a number of ways. We've been able to bring guests like we have today. We've done some gaming earlier this week. I was playing this game called Factorial, doing a little bit of industrial engineering, if you will, on that game. We've been able to build things in real life, models, done some lab tours, and we are collaborating for what will be the finale of this season of Building Stuff. We are going to launch our very own 4-day engineering escape room. That's right. If you've ever been in an escape room, these are fun experiences. But in October in Boston, we're going to have a 4-day engineering themed escape room. And every week we've been ideating with the building committee on Twitch. Whoever joins us, come together with how we're going to do our puzzles, how we're going to bring in certain themes. What is the design going to look like? What are some of the cool quirks and things going to be a part of it? So that when this thing happens in October, not only will our Twitch audience be able to say, hey, we built this together, but you will also be able to participate in it. So make sure you all follow us. If you haven't followed us yet, now is the perfect time to hit the follow button. If you're on Twitch, if you're on Facebook, please do the same thing. And if you're on YouTube, why not subscribe to Building Stuff with Nova, or Nova in general, because we're happy to have you with us. Let's see who else we got in the chat. Shout out to the Facebook audience again. Shout out to the YouTube audience. Let us know where you're checking in from. Let us know where you're checking in from. Of course, Jim Mo is on YouTube, not just the platform, but where in the world? Where in the world are you coming from? Where in the United States? I'm coming to you live from North Carolina, Raleigh, North Carolina to be exact. This is where I've been for a number of years, my education, where I became a PhD in engineering. And so I'm happy to have you all. Just curious to know if anybody's checking in from North Carolina. Hey, future doctors, we see you over in our Twitch family. Shout out to you all. Go ahead and drop where you're coming from as I right now bring my agenda on the screen. Now, for today, here's what we have planned for today. We are going to introduce to you, present to some, introduce to others, a phenomenal chemical engineer that I just happen to be very fond of, have met, got to know a little bit more recently, and that is Miss Camille Smith. Miss Camille Smith is going to come on. We're going to learn a little bit about her profession, her background, some of the cool things she's doing just in general as a STEM professional. And then we're going to get into a little bit of, let's just say, dirty work, if you will, in the area of chemical engineering. We're going to have a lab simulation with our friends at Labsters, a virtual simulation all around, as you've seen in the title, as it comes to regenerating tissue, human tissue. So that's what we got going on. And of course, I'm going to end the stream by telling you what's to come. But instead of waiting to the end of the stream, let me go ahead and tease that right now. We had playing Factorial earlier this week. Yesterday, we had a brainstorming session about escape rooms. Today, we have Camille Smith, who you will be meeting here in just a second. And tomorrow, we're very close, y'all. Tomorrow, we're going to be bringing on our very I mean, not our very first, our second, if you will, lab tour. That's what we're really hoping for. One thing's for sure. Let me just say this, everybody. I will be in Boston. I will, in fact, be in Boston tomorrow. And the goal is to make sure that we have an opportunity on Thursday, tomorrow, to tour, have a virtual field trip, if you will, of the MIT Plasma Center, Plasma Science and Fusion Center. So if you think that's dope, go ahead and drop the number 1 in the chat. You don't have to type a whole lot. Come off chat mute. Type the number 1 if you are digging what we're doing here on Building Stuff with Nova. If you think that is dope and you want to stay tuned for what's to come, drop the number 1. DuckTales, I see the 111 over on Twitch. Drop the number 1 on Facebook. Drop the number 1 if you're in the YouTube world. If you're over there with Jen Mo and Nafis joining us from Philly, shout out to you as well. Happy to have everybody with us today. All right. Future Doctor's with us. Jana, good to see you. Welcome back, our building committee member. Now it's time for you all to meet my good friend, Camille, who is coming to us live and direct. Camille, going to bring you on the screen right now, bringing you in live direct where Nafis is from over on YouTube from Philadelphia, Pennsylvania. That's where you're joining us from, right?
Yes, I'm from Philly.
Oh, man, Philly in the house. Yeah, everyone give some love, give some applause. Let's drop some emotes. If you're on Twitch, drop some emojis. If you're on Facebook or YouTube, at the end of the day, I am introducing you all to my friend, Camille Smith, the chemical engineer. Camille, thank you for being with us today.
Thank you for inviting me. This is so exciting.
Yes. So for people who don't know, before we get into, again, some of the other things we got planned, give us a little introduction in your own words. Give us a little background. How do you fall? What do you do in this STEM world? And who is Camille Smith?
Ooh, okay. So who is Camille Smith? My normal elevator pitch is my name is Camille Smith. I'm a chemical engineer, as you said. I'm also a social entrepreneur and I'm a content creator based in Philly.
Yes.
Chemical engineering is vast, which I think I personally am very excited to actually bring light to everything that chemical engineering can be. And I actually chose it as a major because it's so versatile. So what I do in chemical engineering is just one small facet of everything that you can do with a chemical engineering degree.
Yeah. And I love the fact that you are a content creator because that's where I feel like you and I resonate, because I am here on Twitch, obviously creating content. But as you know, on Instagram and on other platforms, I rock with my company, STEM Media, and you've been a friend of our company, STEM Media, for a while, as we've also continued to create content. And I had the privilege of meeting you in person. Y'all know when you follow people online, it feels like you know them, but then when you meet them in person, it's like, oh man, this is a person in the flesh, right? We met. Where were we at? Was it in Atlanta? It was in Atlanta.
Yes, for NSB, for
Yes.
the national, it was annual.
Say what now?
Conference. I think it was the 50th conference, but it was like a very, very big conference for NSB back in March.
Yes, the NSB, National Society of Black Engineers. For those who don't know what that means, Camille and I had an opportunity to obviously be following each other for a while, finally come in person at this engineering conference and meet in person. And now we have her here on Building Stuff with Nova. So y'all show some love to Camille, who gave us a nice little introduction. Nafis is giving you a round of applause. Future Doctor says, welcome Camille over on Twitch. And we know you're getting love over on Facebook at this time. But we have, as people know, and they've already seen our agenda, we have this virtual simulation that we're going to get into in just a little bit. But when you talk about like your work in like chemical engineering, I'm curious to know, like, what exactly does a chemical engineer do that's different than, like, say, other engineers?
Okay, so that's a hard question. So chemical engineering, as I said, is so broad.
Mm-hmm.
So chemical engineers are really the people that know about processes. So something that I always tell students or people that, interact with me when it comes to chemical engineering is we really are taught how to think through things. So whether that be developing and designing different chemical processes, manufacturing is like huge in chemical engineering. But basically using, chemistry and physics to be able to apply to different industries. So like a lot of people think of petroleum, gas and petroleum is a super big thing for chemical engineers. You can kind of take a hard right like myself. I love biology and biopharma in general. So I work in biopharmaceuticals.
Okay.
So there's so many different things. You can also do material science. So you could do like, things with polymers. different plastics, things of that nature. You could do fermentation, brewing, or making wines, or like things of that nature too. There's so many things that chemical engineering, can be applicable to.
That's true. That's true. As I would say, a lot of engineering, because so many people immediately think about just whatever they're most familiar with, not realizing that there's a whole nother vast area that a person can get into, even within different engineering disciplines, which I think is pretty dope. And you said biopharmaceuticals is your word. What is that? As we get ready to dive a little bit more into the technical stuff, what is biopharmaceuticals? How would you define that?
Yeah, so biopharm is what we call it for short, but biopharmaceuticals is a newer field or a newer industry. What people usually think of is just pharmaceuticals. So that's, your usual, what we call small molecule tablets.
Mm-hmm.
So any given, over the counter, pill or medication that you might take, that would be considered small molecule.
Mm-hmm.
And that's really based on chemistry, organic chemistry in particular. However, biopharm uses things like chemical engineers, biologists, biochemists, to be able to actually work with living things, mammalian cells or animal cells, to produce antibodies. So it's super, super cool. It's been very, very revolutionized. And honestly, a lot of pharmaceutical companies are really taking a hard pivot towards biopharm because that realistically is our future when it comes to medications.
Wow. Wow. If you all think that's pretty dope, I want y'all to let her know in the chat. Let her know that that's pretty dope. And I think there's even more to come, more where that came from. Because Camille, you told me that, you told me a little bit about what you did in preparation for this. And you shared with me that, like, back when you were in school, you were, learning, as we're doing here on this channel, informally even, about a lot of this work through like virtual simulations. And so what I did was, me and the team, we got in touch with the company that you name dropped when we were talking about that, named Lapster. And I'm going to go ahead and share screen here. And we were able to work with the good folks at Lapster. Shout out to Lapster, to work through this demo. You all check this out. We got a demo planned for you all, virtual simulation. Now, I understand that this is not like biofarm in its purest form, but I was trying to get something very similar to what you obviously would have experience with, but something that, maybe relates a little bit to the bio aspect of things. And so what we have here, everyone check it out. Zoeris, I see you. Thanks for shouting this out in the chat. Zoeris said that this is dope. We have a tissue engineering simulation, right? And here in Lapster, what can you tell us about Lapster real quick? Just go ahead. You told me about it before I even try to introduce myself as a structural engineer. What was your experience when you were in school with this whole virtual method of learning?
Yes, Lapster is super incredible. I actually was able to use it for the first time in an elective. I took gene therapy and research methods, and my professor was super adamant about, our school didn't necessarily have the funding to be able to fund a huge lab and be able to show us all these things in person. And that's where Lapster kind of fills that gap, that we're able to do a simulation with that point of view, even down to the importance of wearing PPE or personal protective, equipment within lab, putting on gloves or goggles or things of that nature. So it was a really, really cool tool. I also really appreciated that we could continue to do the simulation until we understood what concept we were doing. So Labster is fantastic, and I'm really excited to be doing the simulation today.
Let me tell you why this is so dope, because it falls right in line with our gameplay motif that we have on Twitch. Shout out to the building committee on Twitch. If you're brand new here on Twitch, now's a good time to hit follow. Shout out to YouTube and Facebook, who we also have. We see you all. Go ahead and subscribe, hit follow. We do this every day at 1 p.m. Eastern. And today we're going to be answering the question, Camille, can we engineer human tissue to treat injuries? That's what I have here. And I'm going to try to go through this. Again, I'm very, very much a fish out of water. But I want to say thank you to you and Lapster in advance for helping me get through this process. Me in chat. I see you, Yana. I see you, Zoeris. We're going to get through this together. Carl, thanks for the love. You're getting a lot of love. I want you to know, Camille. So let's continue let's continue the journey. All right. So here's what we're going to do. We're here in this lab, and they have like this little lab pad here at the corner that we can pull up and look at. All right. So it uses voiceovers. All right, using LabPad is an important way to get the most out of simulation. If this is the first time you played Labster, so it's my first time. It's my first time, but it's not your first time. So I don't know. Should we go with a no? Should we go with no? It's not the first time.
Uh, let's go with yes, since I feel like you and chat might be a
Okay.
little bit new.
All right. Yeah. Yeah. You're right. Thank you so much for considering us. So this is definitely first time as I go through this. All right. Here we go. This lap pad, we're gonna be able to lower it up. Let's continue here. Does it look familiar? Does it look exactly like you remember?
Yes, exactly.
Oh wow. Okay.
Flashback to junior year of college.
Yes. So you said PPE. PPE stands for personal protective equipment,
Yes.
right? We got these different things here. We can use our mouse to confirm and click. Yep, we know how to scroll down. Here's a quick little test. Click on the theory. Okay, so they're gonna. All right, so here's what we see. They're gonna be quizzes throughout this, Camille, and we're gonna rely on you, chat. They're gonna ask us some questions, and when we get to the questions, we'll pause and see if chat can kind of figure it out the answer, and then we'll go to our expert on screen. How about that? When we get to our things, our quizzes. All right, let's get started. Boom, let's get started. And I think we have our PPE on. Shops to Zores. I'm doing research on sales behavior, organic marketplace. Have so many questions. I love it. All right, I love it. Here we go. Dr. Williams. Hi, I'm Dr. Williams, the national football team physician. I'm assuming they mean soccer in this case. I'm thrilled to meet a fine scientist like you to discuss ways to solve problems. Do you have any questions? So for the sake of this education, maybe I'll click on B. What is articular cartilage? Because we want to know what we're talking about. Articular cartilage is the connective tissue that covers the bone ends in freely movable joints. There we go. It enables smooth joint movement and facilitates the transmission of load with low frictional coefficient. Let's see. Should I ask another question? These are just to get us familiar with what we're going to be talking about.
Let's do how cartilage be damaged.
All right. How can our articular cartilage be damaged? Let's go on that. Articular cartilage can be damaged in two ways: chronic joint loading or acute traumatic injury. Chronic joint loading is aware an erosion of the cartilage layer over an extended period of time. Acute traumatic injuries occur suddenly and severely. Boom.
Yeah, so the load will happen to quite literally anybody as they age versus, at least in this case, the football player, potentially could get injured as they're playing.
Yeah. Shout out to our athletes. We got any athletes in the chat? Shout out to the athletes in the chat. This is something that may be relevant to you all. I think we're maybe good now. Thank you, Dr. Williams, for answering those questions. You're very welcome, Dr. Williams said. All right, cool. Let's teleport. I love how we're able to in this simulation, just go to the right next thing. So let's put on our PPE, put a lab coat from the rack and put it on. Yeah. Let's see. And then we're going to go over here to the bench here. Oh, this is pretty cool. Future doctors, I got bad knees for sure. All right. How similar to this lab, really quick, as I'm going through this, how similar is this to the look and feel of maybe your lab where you're at right now?
I'll say this definitely looks very shiny in comparison to our labs. Our labs definitely have been worked in for an extended period of time.
Yeah.
But we actually just did do some renovations. So a few of them are, shiny like this, but then some of them are definitely lived in, if you will.
The benefits of having a virtual lab is that it's easy to clean up, right? You just click, or you just reset, and then everything's back shiny, brand new again.
Exactly.
Yeah. All right, let's see what we got. Dr. Williams explained his problem in treating football player injuries. What are the characteristics of articular cartilage so that it requires treatment by tissue engineering? Oh, this is one of our first quiz questions. All right. Chat, first quiz question. What do you think the answer to this is? What are the characteristics of articular cartilage before we ask Camille so that it requires treatment by tissue engineering? And while you all are answering that, Nova, I have a question for you. If Nova, our fact checkers at Nova, can drop in the chat just a quick little concise definition of tissue engineering so that everybody can refer to it. But I'm curious to know if we on YouTube, where my YouTube people at, where my Facebook people at, where my Twitch people at, do we think we know the answer to this? Are we smarter than a chemical engineer? If I had to guess, Camille, characteristic, articulate conversation issue, I would have to say that it is limited capacity for self-repair. I see what future doctor says, probably can't self-repair. That's going to be my answer. Does that feel right? I'm going to go ahead and click on that. Self-repair. Boom. Self-repair. That's correct. Articular cartilage treatment has been challenging because of the absence of vessels, nerves, and lymph, with limits to cartilage capacity for self-repair. It also handles some of the heaviest loads of the body. All right, cool. Don't worry about it, Yana. We're all learning together. All right. So to make new tissue, we must understand the basics of cell and tissue development. Do you know what the main components of tissue engineering are? Give us just a few. I don't know, but what comes to your mind? Camille, anything pops to your mind as I try to get my bearings here in this lab?
So again, bear with me, chat, because I only took one tissue engineering class in undergrad. But it's really a cross between, again, engineering being like understanding those processes, but also having a good understanding of, anatomy and physiology as well. So even just reference, the cartilage doesn't have vessels, which would make it harder to self-repair. So it's kind of marrying those two and trying to get a better understanding of, again, how we can solve problems with it. We're in the fridge. Ooh.
Yeah, we need to get some like some growth factors, and then what is it? Cells.
Yes.
Right. And then here's what I want to ask you about too, by the way, like this whole idea of scaffold, and we're going to click on that too.
Yes.
So here's some theory here. Approach. It involves combining cells, which is biomaterial scaffolds, and growth factor in vivo applications, which we can isolate cells in the cartilage tissue and incorporate them in the scaffold. The scaffold acts as a template for tissue formation by allowing cells to adhere and develop as tissue. Why is scaffold necessary? Like, why can't we just, why you can't just get like human tissue somewhere else? Like, break down the whole purpose of scaffold if you can.
Scaffolds are really just to help promote, tissue formation. So again, like it said, it provides an actual surface for the tissues or the cells, but the tissue to be able to have like an adequate enough space to be able to grow, whether they need to grow or proliferate, or even depending on the type of tissue, whatever differentiation, it needs to do. So yeah, scaffolding is super important. In vivo, like you can't, because we're working with living things, you can't just, do without certain, like, again, in this case, scaffolds.
Got you. So we got to take this back over to, we got another question. The primary element of tissue engineering besides cells and growth factors is a scaffold. Oh, okay. You were just talking about. In this simulation, we will focus only on creating a scaffold. So we're just going to focus on the scaffold this time. Thank you for breaking that down for us, Camille. What is the main purpose of scaffolds? Hey, we just gave them a little game ahead of time. Chat, what are we thinking? Are we still using pig scaffolding? That's a good question. I do not know. Maybe that's a good question for chat on over there. But we got a question on the screen, you all. What is the main purpose of scaffold? Camille just told us a little bit. We just got some information here on Labster. What do you all think the answer is? Camille? We got act as an extracellular matrix and growth factors, cell source for tissue development, provide 3D structural support. I feel like, 3D makes sense because we're 3D, right? Like we're not like two-dimensional beings around here.
Right.
So, okay, cool. That actually worked out pretty well. Just took a stab at it. They are defined as 3D. Let me go back. Okay. I don't know how to go back to the last question. Anyway, I'll stay here. Numerous types of scaffolds for tissue engineering are available. Which of these scaffolds do you think are appropriate for articular cartilage substitute? Zoeris, is that your answer for this one? Give me another answer. Shout out to Nova. We see you all. The Opal guy, you're here. You are here. Help us out. We're trying to do tissue engineering. what would be your answer to the question, which of the scaffolds do you think are appropriate as articular cartilage substitute? Solid scaffold, hydrogel scaffold, neither of them, both hydrogel and solid scaffold. I know we're kind of just spitballing here. Camille, what should we go with here?
I'm going to go with hydrogel scaffold because, again, materials are also super important when dealing with, human things.
Mm-hmm.
So I feel like a solid scaffold would make too much sense.
So instead of solid, boom. She knows her stuff, ladies and gentlemen. Future doctor says, well, we're going with hydrogel. Hydrogel is it. You're correct. Three-dimensional polymer networks swell in water. It's akin to cartilage structure. Therefore, they are a suitable choice for cartilage substitute. All right, let's go. Hey, I appreciate you all. Hey, listen, I would have said D too, Zoris. Can I be honest with you? But because I have I have the professor here with me, I'm just asking for help. All right, here we go. Today, you will create hydrogel scaffolds using two cross-linking methods, ionic cross-linking and micro-addition cross-linking. One of those is a chemical cross-linking and the other is a physical cross-linking. Before you begin, you can explore the types of polymer we will use today in the rheology workbench. So Camille, I'm just going to try to follow this. If you see anything that looks remotely similar to what you are doing in the lab, please let us know. We would love to hear some of the reality because I'm a structural engineer and I'm pretty much a fish out of water right now.
Well, I wanted to reference a little bit earlier. So we went into, I think that was the incubator a little bit earlier when we got the growth factors and the cells as well, or we went into the fridge.
Yeah.
I personally work with mammalian cells. So there's different types of cells that different scientists will use based off what they're trying to do. But to give a little bit light to biopharm, again, we use mammalian cells, specifically CHO cells or Chinese hamster ovary cells. I've always asked why that in particular. I think it's a little bit odd, but CHO cells are the closest thing to be able to mirror, human cells. From the standpoint that they're able to kind of work as like factories for us to create the antibodies that we want. So when I saw the cells earlier, again, we're not using those cells in this simulation, but that is also something, that you can use whenever you go into a different industry.
Dropping game. Thank you so much. I'm gonna answer this question here, Camille. Crosslinks are bonds. I feel like we just got to answer to this ahead of time. Well, we're gonna in this simulation. We're talking about two different cross-linking methods. And one, I don't remember the name. One was like Michael something. One was physical and one was chemical. So I'm going to go C here. Chemical and physical.
Yes.
There we go. I feel like we're going to pass this class today. We're going to pass this class, though, whereas don't have no fear, chat. We are going to pass today. Let me continue here, see if we got another question. All right. So place a fresh 15 milliliter test tube from the tube box on the rack. Is this how it looks right here? Y'all have like fresh tube boxes? Yes, that's how it comes in?
So what's interesting is depending on where you work, because again, I work in industry, you also can do work like this in like academia labs as well. Mm-hmm. But a lot of the stuff that we use are single use. So we have loads and loads of different tubes, whether it be 15 milliliters, 50 milliliters, so on and so forth. So yeah, we usually have boxes of fresh test tubes ready to be used at any given point.
Yeah.
And of course, it falls on the scientists to replace those as we, go through them for various things.
And then I just put the little thing, break down what I'm looking at here, because I just put like it told me to put the tip thing on the, like, what are all these things I'm using? I'm just kind of following on, but it says it has like a little tip on the end of this thing in my hand.
So, right now you're using a pipette. Pipette tips are basically plastic tips that can, draw up any liquid realistically or solution that you're using. When you put the tip onto the pipette, it creates like a vacuum, which is why you're able to use the apparatus to pull liquid into it. And there's different pipettes that can take up multiple different volumes. So you can have a pipette that takes up 10 microliters. You can have a pipette that takes 100 milliliters. So it really depends on how much solution you're trying to transport back and forth into something. But what's important, at least in this, and they're going to reference this, when you finish with the pipette tip, you have to put that tip into the waste bin, and that's considered a biohazard waste bin. We're very particular about waste when it comes to working, especially in biopharm, but when you're working with living things, it's super important to dispose of things correctly.
Make sure we don't just throw it out on the regular trash. So
safety, very important.
Safety first. So what's this right here? Right now, Camille is saying vortex solution for 30 seconds. What is this whole vortex thing here? Like, what is
vortexing? That? It'll just spin it, if you will. So if you actually click there and place it, I don't know if the simulation.
Yeah, it's spinning. 30 seconds.
Sometimes you have to do that, to mix, your, either if you have like a cell pellet that's, needs to be, I'll say, aggressively or mechanically shaken up, that's when you use it.
So it's supposed to blend it like very well, basically what's going on.
Because again, you can like shake up a test tube, but depending on, what's in the test tube, you'll need a vortex to actually help mechanically, mix it together.
Got it. So moving on here, try to get through this thing. What got you into like even your first interest to become a chemical engineer? Like honestly, chemical engineering in my mind is one of the more difficult ones. What made you choose that to begin with?
I wish I had a better story. I think like a lot of other chemical engineers, I really liked chemistry in high school in particular. Funnily enough, actually, I took honors chemistry, I want to say my sophomore year of high school, and I was terrible at it. I was awful at it. I to the point that halfway through the year, mid-years, I was almost about failing. But I had a really good professor, Dr. Gould. I will always shout her out because she really was pivotal in my, STEM career. Because she would stay after, school and really help me with things until something clicked. And it was specifically polyatomic ions, which feels very weird because, whatever. Does anyone know what a polyatomic ion is?
You understood it though. You understood it that time.
Right. But it made sense. And then everything started clicking. I think chemistry, I was able to kind of logic through, whereas other, branches of science, when I had to rely on memorization, it just took me a little bit longer to understand things. Whereas chemistry, there's a lot of math. You can logic through things. It feels like there's more of a definitive answer. So in high school, I really liked chemistry. I was actually able to take an advanced placement chemistry in high school my senior year. And I think actually my dad just kind of referenced why don't you study chemical engineering? I had no idea what it was. I always joke with him that he set me up because I got to college, majored in that, and then realized, how difficult it was. But I wouldn't change a thing.
Hey, well, listen, if you didn't do that, we wouldn't be here with you today, experiencing this whole laughter experience. Shout out to people in the chat. I see on YouTube, somebody said, Jerry said, Labster, I use this program as a part of my high school environmental science course. Oh, that's dope. There we go. It was a great tool, but became cost prohibitive. Well, I tell you what, that's what I was thinking, cost prohibitive in terms of actually having a whole lab like this. I promise you my high school lab did not look anywhere like what I'm seeing these materials are here on Labster. But shout out again to everybody who is on this journey with us. We see the way that life, Zoeris Nucktails. I'm going to go ahead and go to the workbench, the Rheology workbench, and learn more about these types of polymers that we just got finished working with. Let's see. If I turn the Rheology lock bits, I had to put it back.
I'm going to vortex it again, I think.
Okay.
Because also when you're mixing different things together, depending on their properties, whether that be density or, what have you, you have to make sure that they're mechanically, together.
Oh, okay. But then I got to add, I got to put more on this. I got to take this with me.
And we're going to put it on a slide.
Yeah, okay.
And safety first.
So this is my sample. This is like my little sample thing right here, right? And then I put it.
Incubate it first, I think. That's another thing too.
Go ahead.
Something that you'll learn, when you're working with living things and you're working with, cells. Cells are very similar to people. So for us to be able to operate and feel happy, we need to be a certain temperature. So a lot of times we'll have long incubation steps because they need a certain temperature, a certain set point in order to interact with one another and get the desired result that you want. So like this one only being 30 minutes, I've had an incubation step that was upwards of 2.5 hours. And you just kind of leave the lab, go,
get some food,
whatever
go for a
get
stroll.
food, come back.
You know what I like about this? And for anybody who, again, was like studying this for a test, this is really getting into the details, showing how these monomers are sequentially distributed and either repeating, depicted by numbers 1 and 2, or alternating blocks, depicted in number 3. This depends on the species and age of the seaweed, which I like. Man, this is really, really deep. It really gets deep. It's not just the virtual demonstration. Let's go next. The ionic crosslink only occurs when consecutive blocks of glutaronic or acid, aka G blocks. That's a dope name right there. G blocks. Thus, the amount of G blocks within the alginate will determine the quantity of crosslink established. Okay.
You know, things like this in school really helped me be able to conceptualize what we're doing, because again, cells, when you work with any type of cells are, you can't see them realistically. You know, one cell, when we talk about things at work, we're talking with a unit of per million. So like 1 cell per million, you know, XYZ, right? So this, like Labster, it really gives you the ability to actually see with your own eyes, you know, visualize what's actually happening. So hopefully, you know, you're able to take that concept, you know, into what you're actually doing.
Would you say that, like, what was the most challenging part of like your journey? Was it the technical aspect, or, you know, obviously you like myself, you know, it's not a high representation of people from our community in some of these fields. Like, was it some of the out of class stuff? Was it the in class stuff? Give us an idea of like the challenges you faced, or what were the most challenging things you faced along the way?
It was a mix of both. I do think that engineering in general, specifically chemical engineering, a lot of the material is not intuitive. So you really have to study, the concepts, find resources, you know, like Labster, anything else, to be able to really understand, you know, why things are the way they are. And I think me as a student as well, I, again, really struggled when teachers would just say, oh, well, this is the constant, or, oh, this is the theorem, you know, that you have to know, because I want to know why. Because I'm not going to be able to really, you know, understand it unless I'm able to, you know, logic my way through. So I definitely think some of the material was difficult. Fluid mechanics in particular threw me through a loop. You know, we passed. And definitely, like you said, I mean, I was one of 2 Black women in my class. And there were some days that was, you know, very difficult. But her and I were able to graduate together, which was just as rewarding, you know. So there were highs and lows.
I can totally relate to that. Which ions can aid in cross-linking? Chat, you know the answer to this? What would you go with, Camille?
Calcium.
Calcium?
I'll be telling chat.
Hey, calcium. Ask calcium on here. Sweet. Yeah. I feel like I'm taking the test, and I got, again, like my own personal tutor, like right next to me while I'm taking the test. They should let you do this in school a lot more. Let's continue on.
Right.
See what they got next. All right. So taking my little hydrogel, my little thing out of the incubator. And now I got to make a new one. Okay. Transfer gel to a 6.
Yeah, I was gonna say we're gonna put it on a well plate.
Okay. What's a well plate exactly? Is it like a just one of those things you use to put your samples on or whatever?
Yeah, to put stuff in. It depends on what instrument. I'm not sure in this simulation what instrument we're gonna put the gel plate into. But yeah, you'll see different either gel plates or well plates, you know, used for different things. Like ELISAs, does chat know about ELISAs? They use well plates as well. ELISAs is a type of like assay.
Great work. You have performed the ionic cross-linking using alginate polymers. Now let's continue to the next one. Michael addition. That's a funny name. Michael addition cross-linking. To make hydrogels using this method, 1 polymer with thiols and second polymer with acrylics or vinyl sulfones is required. Okay.
That's another thing too. I feel like learning organic chemistry, like the nomenclature, like how to name things. I feel like you're
I mean, I feel like sometimes the name, like the G block thing was pretty dope. Right. But then like the Michael addition cross-linking, that almost felt like the person's name was Michael and then they just.
But have you noticed that in your education too? I feel like there's just a bunch of random people that just started naming stuff after themselves.
You're right. right.
Like Bernoulli's theorem.
Bernoulli, right. But the names usually are, antiquated enough to like sound like a word, right? But like Michael is like, we all know Michael. Like, Mike, did you do this, Mike? Like,
right.
Let's see. We're gonna keep going. So we're gonna have the Camille something at one point, right? That's what you're gonna name something the Camille edition at one point? Let's see.
Ooh, see, I don't actually know what these are.
You got some nice little camels. I'm just following chat. In case you're wondering, I'm following. Look in the bottom right hand corner. You can see the instructions. It's telling me what to look for and to put it into the tube. So it's just kind of following the instructions. So that's basically what we're doing here. Camille, while we're doing this, let me ask you a question. When it comes to like, what keeps you going? Like, what gets you excited? I know that every job has some things where it's like, like if you had to wait for something to incubate for 2.5 hours and you already ate lunch and you don't have nothing to do, I'm sure that can be a drag. But what keeps you motivated? Like, what do you think about whenever you are like working in the lab and you get excited? Like, what's helping you continue to move?
Yeah, so 2 things. One, I'll say at least like when I'm in lab is realizing that I'm quite literally making medications for patients to improve their quality of life. That's something that like, I don't know if anyone in the chat can agree or empathize, but I have like the triple threat. I have asthma, I have eczema, and I have terrible allergies. So all my entire life, I've had to have like over-the-counter medications, EpiPens, inhalers, like things of that nature, right? And those objectively improved my quality of life. I was able to, navigate the world even with, different things that I was experiencing because of, the different medications, that I had at my disposal. So to be able to be in lab and, innovating new medications, is crazy, so even through the long incubation periods, you kind of go back to, your moral compass, if you will. Like that's what's really driving me forward, especially for someone that looks like me to be behind like the innovation of the medication. I think is also very cool. It's something like I really proud on.
So it's really that why, even beyond just like the actual details of what you're doing. Because it's not like, oh, I like opening tubes and putting them in the rack and putting dry. Like it's the application and the bigger meaning to it that keeps you going.
Right. Yeah. And I do think that like my company in particular does a very good job of trying to ensure that we don't forget that why, like why we're working so hard, why we're trying to, get data or generate data and, do different experiments. Because we're literally improving people's quality of life, and being able to interact with clinical trials, seeing what the readouts are and like so on and so forth. It just makes it feel much more worth it, especially on harder days.
I love it because I'm doing this. This is fascinating to me. And I'm back here at this incubator, put another gel caster in there for 15 minutes. And like, it's novel to me, but somebody like you're in the lab like very often. Oh, another animation. It's pretty dope. You all go ahead, feel free to follow along. I'm right here with you. But Camille, on a day-to-day basis, what are you doing like day-to-day in the lab? Like, what are you doing day-to-day?
So it really depends. So if I have an experiment, I'm prepping for it, I'm preparing. So again, I personally work with mammalian cells. So we use, we have to do a lot of planning, a lot of like experimental design to know or to generate the data that we really want. So it's a lot of meetings to ensure that everyone's on the same page. It's a lot of requesting of different things. So for inland sales, we have to use media. There's different types of media that you can use in industry versus in, I'll say like academic labs. We also use, I actually was able to do my first external talk recently, and I was talking about the importance of glucose. Glucose is the main carbon source for, cells. So I already referenced that cells are basically humans, but just smaller, right?
Smaller, living
like us, we need to eat, we need to be at a certain temperature, we need air, we need certain things. Cells need the exact same thing. So really coordinating a lot of that. And then day to day, being in lab, sampling, seeing how the cells are actually doing. We have a variety of different instruments that can test, how many cells are alive, how many cells, are dead, the general viability, how big the cells are. And to me, seeing different, measurements, that tells me as a scientist based off of, my previous work, where we are with the experiment. And then outside of that, a lot of meetings.
As we all love. The best part of all of our jobs, meetings.
Visualizing, all that good stuff. So it's been cool. It's been really nice. I've been working as a chemical engineer and as a scientist for now 4 years, which is really cool.
Dope. Ever since you graduated. And I'm going to try to chat. We're going to try to keep moving here, even as I got more questions for you, Camille, that I want you to answer, even as we continue to go. So if you see the tab move faster than you all can read, that only means that you got to take some time to get in the lab here on Livestream. Right. You get a chance, chat. But Camille, let's talk about now. We see the Opal guy. Jerry said this program was huge. If you have any questions for Camille, feel free to drop them in the chat. Zoairs asked a question earlier. Have you ever done anything with calcium hydroxide? Does that come across your work at all?
Not in my current job, but I did do something. I've done an experiment with calcium hydroxide before, but that was, I think, in orgo, which again was a few years removed at this point. These are the scientific questions.
I know, that was definitely a scientific. Shout out. We got a smart crew here, by the way, on Twitch. And for those who are just tuning in again, this is the Building Stuff with Nova. We do this every day at 1 p.m. Eastern. This week we have an incredible lineup. Right now, we're going through this simulation with chemical engineer Camille Smith. That's all about regenerating human tissue after injury. We're trying to save this football player, this soccer player, the cartilage. We're trying to save it so that we can get them back on the field and win. Yeah. Which of these cross-linking methods generate relatively non-permanent cross-links? Is it Michael addition? Michael addition, that's what you said? Let's go Michael addition. Yeah. This type of cross, yep. Oh, try again. All right, let's see. Is it ionic? Let's go ionic. Because those are the only two that they talked about. Ionic cross-linking. We should have known not to trust Michael. He naming stuff after himself. Michael names stuff after himself. He misleading us here. All right, let's keep going. Continue. All right. Gonna try to again get this done here. So chat, sorry if we don't slow down on every question. What is your life like outside of the lab? Break it down. I know you got a lot of cool stuff going on. For people who don't know, Camille is very active on social media, Instagram and across other platforms. And you have a pretty dope lifestyle outside of the lab too. Tell us a little bit about what you like to do, Camille.
Yeah, so outside of being an engineer, I'm also a social entrepreneur. So I have an organization called STEM Social, which I consider a hub for aspiring and established Black STEM professionals. So we help our audience in 3 different ways. A, we are currently in 15 major cities around the United States, and we host different social networking events. B, we also have a personal and professional workshop series to help upskill our community. I actually had the doctor himself, come and do a talk, which was super amazing. And then lastly, we also help our community by, having scholarships for current undergraduate and graduate STEM students. So we were able to give out our first round of scholarships at the end of last year to 3 different students, which was super, super exciting. So STEM Social definitely takes up a lot of my time. It's growing very rapidly, which I'm very happy about.
Yes.
And then I also do content, as was referenced. I don't even know what niches I am anymore. I'm the niche. It's me. But I talk about, being in STEM. I talk about, early career advice. I travel a lot for STEM Social and also personally as well. So, a lot of my travels I talk about too. And then also I'm a sucker for skincare. So I also do a few skincare videos too, talk about my new favorites. But yeah, between the two, it's like I'm living a double/triple life.
You're doing a lot, yes.
Founder, but also content creator. I don't know.
You are a travel, what is it? They call it travelista as well. Is that an actual name? You
a nomad.
You're a nomad. You travel. We see you all over the world. Y'all gotta follow. Go ahead and drop your handle as well. I am Camille. Yes. Feel free to drop your handle as we continue here. Rheology as a method suitable for characterizing. Okay. The yield stressor gel. Let's keep going. We're about 70% done here. 71%.
We got it.
All right. Yeah. Zoe is showing some love. Go Camille in the chat. Thanks for the love. We see you all on YouTube as well. Change the parameter of the modulus. Now I know a little thing about this actually. Please try a different cross-linking. Are you ready? Yes, we're ready. So the modulus is similar to some of the stuff I'm doing as a structural engineer. Let's see. Physical. Let's see, which one is this? Based on the data, cross-linking methods are very strong. Michael Edition. We'll trust Michael this time. Go ahead, Michael Edition. Yep,
there we go.
All right, Michael, we trust you this time. Let's continue. We have about a little more time. Let's see. When it comes to your, again, STEM Social, really quick, like how did that even start, Camille? Like how do you even start getting into that type of work?
So it actually started with content creating. So I started creating content on TikTok in particular in 2020. And again, I was talking about, being an undergrad, different story times, things of that nature. And then my comments started to become a lot of people saying that, they also were experiencing, a lack of representation in either their departments or, where they worked or so on and so forth. So then a lot of people started asking me to organize events, if you put on an event, we'll show up. But sometimes I love the internet. However, sometimes people on the internet will just say things and they're not necessarily truthful. Yeah. So I was a little bit hesitant initially, yes. But they were very persistent. So I decided to have an event in Philly. It sold out. And I was like, okay, the event went super well. I met a lot of cool friends. And it's really just been going from there. So I've technically been hosting meetups for the past, I'll say 1.5 years, 2 years. But STEM Social recently hit its 1-year anniversary in June.
Congratulations, STEM Social. Shout out to the STEM. What do you call the STEM Social group? We like to call on Twitch a building committee. At least that's our name until further notice. Do you have any name for the STEM Social community?
I don't know. I don't think so. Oh, I'll have to ask my team about that.
Yeah.
What are we going to call it? That's funny. Yeah, no, I don't know. Because I don't usually name things. Like sometimes people will name like their cars or like name,
right.
And then I don't really name anything. I will like personify things, personify, I believe.
Mm-hmm.
So like I'll talk to my cells in lab and I'll like ask them how they're doing. Or like my plants. I'm a plant mom. So
you're a plant mom? So you name your plants?
No, but I just talk to them as if they're people.
So you personify them like a person without a name.
Yes.
Maybe that's a mark of genius. I don't know if that might be a mark of genius, chat. Camille might be on to something. Maybe we can personify the stuff around us. Maybe that'll give us the same type of genius she has.
Listen, it works. All right, well, we're almost 80% of the way. Okay. So we have cross-linking. Let's go meet with Dr. Williams.
Yes, Dr. Williams.
So what are you doing?
Scaffolding, another question. Piece of scaffold.
What's the ionic?
What is it? Alternate and ionic. Okay, yeah. C.
Yeah.
I feel like the professor's gonna take my paper soon. All right.
Right.
Let's see. Take the phone call on the hollow orbit. Look at that. Look at that. We got the little. I need to review. I think I understand the cross-linking part. I understand the types of polymers and cross-linking. We've got a lot of that.
Mm-hmm.
So we basically just need the ideal mechanical properties of cartilage replacement are not known. More studies are required. The effect of hydrogel stiffness in cartilage tissue engineering. The hydrogels must be stiff enough. Yeah, this is what I was going to say. This is similar to structural engineering for us. It must be stiff enough to support physiological loads or they'll collapse. The rate of degradability should mirror the rate of new tissue. So we want it to basically behave as similar to actual tissue as possible. When we put it in, we don't want it to be more stiff or less stiff. Let's continue. Which is not.
Ooh, not.
Which one is not? Let's see. Polymer concentration. Dr. Williams is not. This maybe it's the site of the. Is it the site of it, or is it the top?
The site.
Yeah, the location. As long as it's acting the same, right? D.
Right.
Bada boom.
On exams, when I'm like, okay, not.
Yeah.
So which ones are?
Exactly. Let's continue. We're almost done here. What will the hydrogels become? Hydrogels become when the elastic modulates get higher. The higher the modulus, the stiffer it is. I'm gonna go stiffer. That's where my structural engineering background comes in. Boom.
Yeah.
Wait, no. Am I wrong? Oh, wait. Am I
that's
wrong?
right. That's right.
It's wrong.
No,
Okay.
You're right.
Okay.
Yeah.
All right.
Structural engineer.
I'm looking forward to say genius though. I wanted to say genius, Camille.
Right.
It says to me to other answers that you gave. I want to get the genius. Maybe I need to start talking to things like it's a person, and then I can get the genius.
Right.
I can get the genius gene. All right, great job. You've explored natural and synthetic polymer today. In the future, you could explore more about combining synthetic and natural polymers, creating a hybrid polymer. The synthetic polymers provide tunable physical properties, while the natural polymers provide specific biological functions. For example, cell adhesion. I'm sure this knowledge will help to treat the injured knee of the football player. Chat, I think we did it. Let's show some love. Let's drop some emotes. Oh, we got one more question. One last question. Do you feel you gained relevant knowledge during the simulation? Chat, I'm gonna let you all answer this. I think I gained a little bit. I even was able to tie it into my structural engineering knowledge here with the help of Professor Smith on my shoulder here on Twitch. So we're gonna go agree, chat. Type agree if you agree, or completely agree if you completely agree. I completely agree. Somebody said I wish they had somewhat agree. Look at our score, 98 out of 10. Hey, Camille.
Yeah.
Am I ready? Like, will y'all hire me in the lab? Like, is that where we are now? Am I ready for a job? You gotta let me know. How much y'all paying? What's the offer? Y'all, it's Camille. As we switch our screens here a little bit, Camille. First of all, how was similar to this a little bit? Any other comments on just like the virtual experience we just had versus the real life experience? Anything else come to mind?
Yes, I will say that as we were doing the experiment, pipetting, there's a lot of repetition. So it's kind of funny how like ergonomics are like very important to scientists, and making sure that because we're doing so many repetitive motions, like we're stretching, we're making sure that we're walking, we're not like sitting in the same positions, because it can eventually over time, put some wear and tear on your body.
Right.
So yeah, a lot of pipetting. That's honestly why well plates are nice, because if you're able to get a serial pipetter, which is a pipetter that has like 8 or 10 tips, you're able to pipette a bunch of stuff at the same time, but you don't always get that luxury. But no, very similar to what I do. Yeah.
I don't think about it. I think it's one of those things where it's like, you can have workout specific to like certain things. And I didn't even think about like just the physical tiredness of what, for me, it was just moving the mouse, right? For anybody doing this, it's just clicking. But like you get tired of doing the same thing over and over again.
Also, the last thing I'll say is that we were on our feet technically the entire time while we were in simulation. We have to wear very comfortable shoes in lab. There's not always going to be chairs. Also, we had the incubation periods, but they weren't really long enough to take off all your PPE and go grab food or go grab, water. So you have to be very cognizant of, timing out that stuff at work.
For sure. Again, shout out to chat. Shout out to everybody hanging out with us on YouTube, Twitch, and Facebook. We got a 98 out of 100. I think at this point, we're all ready to be chemical engineers. At least in this very specific simulation, we're ready to be chemical engineers. Camille, we really appreciate you. Can you give us some final words of wisdom to everybody that's hanging out with us? Of course, we want to make sure that everybody knows where they can find you beyond this, but give us some parting words of wisdom before we say goodbye to you.
Two things. First, fail forward, fail often. I think honestly, I would have gotten to the point that I'm at, as a scientist much quicker if I was just not afraid to fail. You are going to fail. That's a part of being in STEM in general. So don't be afraid of it. Just run towards it. And B, if you're in, school right now, you're considering doing school, take something that you have some interest in, but maybe you don't have, the full foundation for. In school, I was able to minor in ethics, actually healthcare ethics, and I absolutely loved it. It's honestly one of my favorite things from, my undergraduate career, because I was able to have more of like a holistic view on the world and also like what I was doing as well. So always try new things and always fail forward and often.
That's it. There it is, y'all. Y'all heard it from chemical engineer Camille Smith herself. Please drop some love in the chat. Give her some emotes, some hand claps. That's right, YouTube. Show her some love. Let's thank her for hanging out with us. Yana says Zoeris needed this. Facebook, go ahead and shout some love out to Camille. Camille, I am super happy that you were able to hang out with us today. Chat, I want to let y'all know what we still got going on. We have breaking developments, if you will, breaking developments. So I told you all at the beginning of the stream, when Camille, before she got on, that we're trying to go to the lab tomorrow. Well, we're going to have to make a change. We're going to make a change. The team is telling me we're going to have to make a change. So we won't have a stream tomorrow. Instead, we're going to have our virtual lab tour on Friday. There you go. You see it on the screen right now, that on Friday, and not tomorrow, Friday, not Thursday, we're going to have our virtual lab field trip tour of MIT Plasma Science Lab. So mark that on your calendars. Tomorrow we'll be hanging out. Maybe you all can take some time to review what you learned today on the back end of our exam. But I want to make sure you all had a good time today as we went through this virtual experiment here with Camille. I'm super happy to have everybody with us here on Building Stuff with Nova. So make sure you hit follow if you haven't on Twitch. If you're on YouTube, hit subscribe and then join Twitch. Hit follow there. If you're on Facebook, make sure you hit follow. And why don't you continue to join us every day right here on Building Stuff with Nova. Every day is stuff for tomorrow because we're moving that tour to Friday. But until next time, you all take care. We will see you later and keep building. Peace.